What we've been looking at with evolution is the process of evolution. And we've been looking at how does it happen and what are the mechanisms. And we've really been looking at the how and the why. But we do also need to look at the what and then question the what. So the first question that we have is, how is evolution observed? And one of the things that we'll see is, for example, we will see diversity. But sometimes we'll also see unity. And so when we're looking at patterns of evolution, we want to be aware that we might see one or the other, we might actually see both. When we want to actually be able to observe it firsthand, we have to think about, well, where am I going to see evolution? If I want to see evolution, where am I going to go look? Well, we have to keep in mind that evolution is, if we define it, remember, it is the change in the gene pool over generations. So one of the things we have to pay attention to is the length of the generation. And so organisms that have a long time between generations, so for example mammals, we're going to see that it's a lot harder to observe evolution when there's a long time between generations. So insects and bacteria and other organisms with short generation periods. These are the organisms that are the easiest to observe at natural selection and evolution in their population, because I need to track a population over multiple generations. We will see this in artificial selection, so your book does a great job of talking about the domestication of wolves and foxes. So if you're a dog person, this is actually a really interesting article. But the idea of artificial selection is usually going to be found in agriculture and farming. And this is where the breeder is going to select and a lot of times record. One of the interesting situations that would have been a great opportunity to study natural selection would have been Mendel, if he had actually kept track of allele frequencies and the idea of purposefully choosing who mates over time. And so here comes this idea again of allele frequency. So if I want to see evolution, first I can look for organisms with short generations, or I can look for artificial selection, or I can track the allele frequencies. A couple of things to remind yourself of here. The Hardy-Weinberg equations, the Hardy-Weinberg equilibrium conditions, meaning large population size, random mating, no gene input, no natural selection, those sort of principles. And then we also have the 5 agents of change. And so these are all going to help us kind of track allele frequencies and therefore see evolution. So we can see it firsthand, we can see it happening. We know that evolution is still happening today. But what if we want to look at evolution that has occurred in the past? What if we want to rewind and look at where we once were? And so this is where we start to talk about what sort of evidence is there. This is the big question that a lot of people bring up. Well, what's your evidence that evolution has occurred in the past? And one of the very few tools that we have is looking at preserved remains or traces of an organism, and these are known as fossils. Most people think when we say fossil of bone, but it can be the remain of the organism. It can actually be just a trace, so sometimes even a footprint or a nest itself is an example. We do need to talk a little bit about fossils and why they are a very imperfect source of evidence for evolution. They are imperfect for a couple of reasons. One, there's a pretty large rarity of formation, meaning not every organism leaves a fossil. actually need anoxic conditions, meaning no oxygen, and this will stop decomposition. So if there are decomposers like bacteria or fungi, then they're going to break down the organism, including the organism's bones over time. And remember, we're talking millions of years in this geologic timescale of Earth. And so we do have to realize that not every organism leaves a fossil. Not every fossil is going to be in the condition to give us any information. We might find just one bone of an organism. So examining fossils is a pretty challenging field to be in. However, and so what this leaves us with is something, called an incomplete fossil record. And we're going to talk about that in just a second, where we look at how did we jump from ancestor 1 to species 3. We'll get there in just a second. We do want to realize the advantage. We can actually get a pattern of organisms over time. So what we are looking at here are levels of the Earth's crust, and we're actually able to see patterns over time. A real casual analogy that I like to use is the idea of your laundry basket. And when you change clothes, if you put your clothes in the laundry basket, you actually have a record of when you wore what clothes. The older the clothes are down towards the bottom. The more recent or the younger clothes would be up more towards the top. Of course, this only works if your laundry basket stays intact and if the laundry actually gets into the laundry basket. So it's an okay analogy for the idea that we have an incomplete fossil record, but it does reveal some patterns of organisms. And so the patterns is really this idea of relative dating. I have a relative idea. I can make predictions that ones at the bottom are older than ones at the top. And so relative dating means getting a range or an approximation of the age. And so it's more using these ideas of younger and older than exact time frames. What we have been able to do is use science, use chemistry, to actually do something called the radiometric dating. And radiometric dating is going to give you a pretty precise date due to something called isotope half-life. And we want to take just a second and try to understand how we get these ideas of 4.5 billion years old. When we think about an isotope, keep in mind that an isotope varies. It's an atom that varies in the number of neutrons. And so what we'll see is radioactive decay. The idea that we're actually going to lose protons over time, and as I lose protons, I'm actually going to change the type of molecule that I am. We're going to see differences in the proton, neutron, and electron equations. So that's kind of what radioactive or radiometric dating does. It looks at the atoms and how well they were held by the nucleus. The key thing is that we're actually able to measure how long it takes for a sample to change from one to another over time. And so uranium U-238 has a half-life of 4.5 billion years. Meaning if I have a sample of uranium-238 and it changes into X, we don't even need to know what X is. But they might give you an equation where they say we have sample A that turns into sample X, and it takes 4.5 billion years for half the sample to change. So this means that if half the sample has changed, half-life, that 4.5 billion years have occurred. Well, what if I tell you that a quarter of the sample is left and I've lost 3/4? That doesn't mean 3/4 of this time. That means that I've had one half-life And that the half that was left also got cut in half. And so really that's 2 half-lives. And that would be 9 billion years. So if I have a quarter left, that means that I went from 1 to a half to a quarter, and that is 2 half-lives, which would equal 9 billion years. So this all comes down to chemistry and being able to analyze atoms and look at how they change over time. And that's how we get precise ages of fossils. So we're able to look at the atoms that are in that fossil. So again, we're going to keep in mind that the fossil record is actually incomplete. There are gaps in the fossil record. We have to keep in mind preserving the fossil and recovery of the fossil. But the main idea is that if I see organisms and I'm able to see a pattern in that organism over time, I can use that as evidence for evolution. So we just want to look a little bit more at this idea because this will help us develop a time frame of evolution. So there are 2 main time frames that we're going to talk about when we discuss evolution. And so of course time frame is going to really mean over time. How quickly? What is the rate of evolution? And so we're going to see 2 theories, and it is not versus. It could be one for a period of time and then another. It could be just one, it could be just the other. It's not a one or the other, so we do have to be careful with the word versus there. Gradualism is exactly as it sounds. It's the accumulation of small changes over time. Gradual evolution. And so the idea that you can see here in this picture is that over time we would see gradual changes. Now interesting, and so you can imagine, well, there's one mutation, and then there's another mutation, and then 1,000 years later there's another mutation, and 1,000 generations later there's another mutation, and eventually some stayed the same and some changes, and that's how we have this change. Well, if you've really looked at this picture, these 2 sidebars are the same. We're looking at the same fossil record. And so we're asking ourselves, did this fossil record occur through gradualism? And part of it is just not preserved. And this is why the fossil record's a little tricky. Is it that it happened slowly and I just don't have any evidence? And that's gradualism? Or maybe I do have some intermediate ones? Or is it something called punctuated equilibrium? Punctuated equilibrium is where we have long periods of no change. Followed by a rapid change in the gene pool. Punctuated equilibrium. The idea of punctuated equilibrium would be that either we had a pretty drastic mutation or that we had some sort of environmental condition. So remember the idea of genetic drift? Like a bottleneck. Maybe everyone died except this variant. Or a founder effect. Founder. So we've got these 2 different theories of how quickly evolution can happen. Is it the accumulation of small changes over time? We do see evidence for that. Or is it a rapid, drastic change followed by periods of stability? Keeping in mind that if I have a dramatic environmental change, a lot of times that is going to cause some pretty drastic changes in the gene pool as well. And that's where we'll stop. We're going to go on next to talk about anatomy as evidence and what are the problems with looking at, for example,